Gene regulatory networks. We'll be looking at how the gene regulation works, gene regulatory network, and the implementation. Gene regulation. Regulation of gene expression includes a wide range of mechanisms that are used by cells to increase or decrease the production of specific gene products like protein or RNA. In the transcription process, the regulatory proteins in the human body can bind to certain regions to increase the rate of transcription for enzyme RNA polymerase. These proteins are positive transcription factors. There are also some regulatory proteins which decreases the rate of transcription. They are called negative transcription factors. These regulatory proteins bind to a regulatory sequence. So, without the regulatory proteins and the sequence, we couldn't make the RNA polymerase and the protein. Gene regulation in prokaryotes. One of the key players in gene regulation is RNA polymerase. RNA polymerase starts the transcription process, but it doesn't know where to bind. So, it needs a promoter, which is a sequence of DNA where the RNA polymerase can bind to. When RNA polymerase gets attached to the promoter, it expresses the gene, which makes the mRNA, and that eventually makes the protein. Another sequence of DNA called operator is there. Operator is a part of DNA where the repressor can bind. Bounded repressor blocks the RNA polymerase, so no mRNA can be made. For example, in lac operon, if lactose sugar is around, bacteria want the enzymes to be made so that they can use them to break down the lactose sugar. If no lactose are present, then the repressor binds to the operator, which blocks the RNA polymerase, so no mRNA can be made. If lactose are present, they bind to the repressor. This changes the conformation of the repressor, so they can't bind to the repressor operator. So, polymerase makes the mRNA. In the translation process, mRNA is converted to the protein. Gene regulation in eukaryotes. In eukaryotes, it works with the help of transcription factors. The transcription factors will allow the attachment of the RNA polymerase. In order to make the gene, DNA will have to fold with the help of transcription factors. When the gene folds back, it activates the RNA polymerase to make the mRNA and the protein. There are no repressors, so here the switching on and off of the promoters occurs during the folding of the DNA. Gene regulatory networks. Gene regulatory network displays the relation of transcription levels between different genes. GRNs visualize the relationship between different genes by representing the connections as a graph. Genes can be viewed as nodes in the network, with the input being proteins such as transcription factors and output being the level of gene expression. The complete network of gene interactions resembles a roadmap. It's a roadmap for us to understand all the effects that can occur when the levels of one gene are changed. By modeling gene regulatory network, we get weight matrix giving the output of interactions between two nodes. Now, using these output weight matrix value, we can calculate the value of gene expression required for the particular change in cell. Let's look at the implementation of the code. We have used the concept of machine learning to make the gene regulatory network here. sklearn is a library to use the machine learning classifiers. We will understand how the machine learning concept works in a moment. First, let's see how the dataset is given. We are given two input files. One contains the names of the different genes of yeast, and other one contains values of different features of a particular gene. We transform this .mat file into a matrix using data frame method. The combined matrix has features of genes as columns and their names as rows. We also created empty matrix which we will be using later. In the loop, one by one, we select one gene as a target gene, and for that target gene, every other gene is an input gene. Here, X is a set of input genes and Y is a target gene. Now we have the data in the form of a matrix. As shown in the figure, each column is a feature of the example data. In our case, as we are considering time series dataset of yeast, these features are the levels of gene expression taken at time t. We have taken data of 6178 genes at different times. Let's see how the machine learning works here. First, we collect the training data. According to this training data, we train the classifiers, and then these classifiers make predictions. Here, we are using decision trees as our classifiers. The training algorithm is included in the classifier object called fit. We have used a random forest regressor algorithm which uses decision trees. Let's understand how the decision trees work. To make it simple, let's understand it for a tuple xy. To construct a decision tree, we construct a binary tree and minimize the error in each leaf. We are given a data distribution here. For each target gene, we have a set of input genes. Here, X is an input gene and Y is a target gene. First, we split the data on an x-axis at different levels randomly. Now, we make a decision tree as shown in the figure for each partition region. We calculate sum of squared error, which is a mean squared error at each split point between the predicted value and the actual values. The variable resulting in minimum error is selected for the node. This process is recursively continued till the entire data is covered. In our case, we have formed 200 decision trees randomly from our dataset. These many decision trees lead to a random forest. We have this matrix. Now we create random subset of that matrix, and hence it breaks down the whole dataset into smaller and smaller subsets, like we had shown in the previous distribution graph. Each sample subset is a decision tree. We repeat the same process on each decision tree by creating random subset of it recursively. In the code we can see we have created an object of random forest regressor with the number of trees to be 200 in the forest. Now, the fit method builds the forest of 200 trees using the input as x and y. As we know, these decision trees are the training models, so from these 200 decision trees, we get a weight matrix and feature importance matrix. The feature importances matrix consists of a mathematical relation between each genes in terms of gene expression levels. Now, we put these values in the Netwrok matrix, which is then converted to a CSV file.